So to do this, it's not quite true we don't have to do anything, but we don't have to do an awful lot. We have to take things out of the media to try and make the stem cells become retinal pigment epithelium cells. So what we do is we take our stem cells, and before we had to take care of them every single day. Here we have to a little bit forget about them. We have to put them in a plate, and we have to let them grow as much as they want until they fill the whole plate. And when we do this, we see little black spots appearing on the plates. They can be seen here a bit higher. What we then have to do is, under a microscope, cut out these little black spots mechanically and put them onto like a jelly that contains a lot of factors, growth factors that it needs, that the cells then need to grow. And when we do that, these cells become very different. That we can see now they're a little bit hexagonal and they're pigmented. So they have a characteristic that was typical of RPE. But again, it was like the stem cells. It looked like stem cells. Are they stem cells? So here it looks like RPE. Is it really RPE? And so again, we had to do a variety of tests. Now, the RPE cells have a morphology, a very specific morphology. For one, they stand up in the body. They're standing. At the top, they have like hair. It's called microvilli. This is at the very top. At the bottom is their nucleus. And in between, we have the cytoplasm with all the melanosomes, which is what makes them dark, makes them pigmented. So the first thing we did is we used electron microscopy to look at these cells, to see what is their appearance. And with a scanning electron microscope, where we look over the surface of the cell, we can see the little hair-like structures that they should have. When we cut the cell, if you like, with a transmission electron microscope, so now we can look inside the cell, what we could see is, on my pointer, at the bottom here is the plate, and at the bottom of the cell is the nucleus, and at the top is the little microvilli, the hair. So the cell is indeed polarized, it's standing. And in between we have lots of melanosomes, which give them their pigmented color. So the next step was, they look like RPE. Do they have the proteins that tells us they really are RPE? And there's a lot of proteins that we know are present in these cells. So here I'll give you an example of 3 proteins. So what we do is we label proteins in different ways. So here we've labeled a protein that's in the cytoplasm. It's a member of the visual cycle, which takes place in the retinal pigment epithelium and the photoreceptors, and it's important for generating the visual pigment, which helps us see. And we've labeled here a protein in green, which shows this protein is present. Here is another protein. It's a little bit hard to see. It's in red. It's a protein specific to the microvilli, to the hair. And here again, we have this protein in our cells. And we looked at another protein, which is situated at the junction between the cells. So the epithelium, retinal pigment epithelium, is what we call tight. The cells all sit very, very close together, and nothing can go through, no liquid, nothing. And this is actually, this tightness is performed by something called desmosomes. It's just a collection of proteins. And this red marking shows us that these proteins are there. So we knew now we had a real retinal pigment epithelium in our plates. The question is, does it function? Does it still work? Can it do in the plate what it does in man?